Hey, here's what we're doing. Today we're going to continue to plug along Chapter 10. And actually, if I can get through the information today I want to get through, I think tomorrow is going to be a lab day instead of a worksheet day. We're going to do a lab, however, that depends. Tomorrow it'll be a microscope lab day, and remember you're dealing with very expensive equipment, so as long as I can see everybody kind of come in and look like they're ready to work, then we will do the microscope lab. So, at any rate, what?
You said it depends last time, another classmate didn't ask.
Yeah, that is true.
No.
Yeah.
No, I actually trying to design your lab so that we're not roaming around the room because there's so many people. That's kind of how I'm trying to modify the labs. Not necessarily not do them, but
Cool.
change them around. So we'll see if we can do that. Okay. Let's see. Actually, the last thing we talked about, we started this new chapter and we basically came to the conclusion that cells are better off if they are small. Easy. Okay. Cells are better off if they are small, because if they are small, then they can efficiently move substances in and out of them. Okay, that makes sense actually. So then this chapter is all about cell division. Okay, so cells are going to divide in order to kind of keep that small volume and shape to them. Okay, so the first thing we did when we talked about the last time we spoke was, I think I had you guys draw a pizza. Okay, do a big circle. And we named the parts of that pizza. We said when it comes to a cell, in other words, when it comes to a cell is going to spend most of its time growing and doing some other things, but really it's called interphase. Interphase is the phase where a cell spends most of its life cycle. And at the very end of that, it may decide to divide. Not all cells divide, but I'll get to that another time. But for the cells that divide, it represents a very short time of their life. And cell division is a two-part process. Mitosis and then cytokinesis. Okay, both of those together are cell division. Mitosis is what we're going to talk about today. That's the word of the day. Greenhouses on anymore.
I seem more tired than usual.
All right, mitosis is one part of the cell division process, and the definition of mitosis is just division of the genetic material. Okay, that word, genetic material. Here's kind of what gets everybody kind of confused. Genetic material has a lot of different words. Genetic material is a very general word, but throughout this chapter and throughout this process, genetic material is referred to as chromosomes, DNA, chromatid, sister chromatid. There's like all these names that have to do with the same stuff, the same genetic material. And all those different names have to do with basically at what step of the cell cycle, what stage of the cell cycle you're at. And then it has like a different name referring to the genetic material. So that's where everybody gets confused. So that being said, we're going to that's one point to kind of keep in the back of your head. We talked a little bit about some of those words the last time we spoke. We talked about chromatin being this thread-like version of genetic material. Then we talked about chromosomes being this packaged unit condensed version of genetic material. A sister chromatid. Did we bring up that word? Anybody remember what a sister chromatid is?
I don't think
we didn't talk about that one yet. Okay, we'll get to that today because that's actually kind of we'll get to that one today. I see. Well, today we'll talk about something called a sister chromatid, which is still genetic material, but it's in a different form. So we'll get to that. Before we get to those details, big picture. What does mitosis result in? The cells that are made by mitosis, daughter cells, daughter cells are going to be genetically identical to the parent cell. Yeah, okay, good. Okay, in other words, that's a fancy way of saying, you know what? If you start off with a parent cell and you know it's going to divide. It'll divide and make two daughter cells. Those daughter cells are going to have the same genetic material as a parent cell. That's all that's saying. They're going to be a clone, in other words. Another important thing about mitosis. Oops. The daughter cells will have the same number of chromosomes as the parent cell. Not only will the cells have the same genetic material, but they're going to have the same number of chromosomes. Okay, I have a picture here. I have a picture of a cell with a 46 in it, and that basically is telling you if you start with the parent cell here that has 46 chromosomes, by the time mitosis is done and cytokinesis is done, you will have two cells and they will also have 46 chromosomes. Now, that's humans. Humans have 46 chromosomes. So when a human cell undergoes mitosis and cytokinesis, you're going to make two new cells with 46 chromosomes. However, not all organisms have 46 chromosomes. There are some organisms that only have 8 chromosomes. There are some organisms that have more than 46 chromosomes. We don't have the most. Anyway, if a cell with 8 chromosomes undergoes mitosis and cytokinesis, it'll result in 2 cells with 8 chromosomes. That's kind of what that says. Again, here's the main picture. When a cell undergoes mitosis and cytokinesis, you're making the same cell and you're making the same number of chromosomes. You're making a clone. Okay. Also, finally, the type of cells this happens in occurs in something called somatic cells. Now, that's a fancy word. Somatic cell is any cell other than eggs and sperm or their precursors. What that means is not all cells are going to undergo mitosis. It's only reserved for somatic cells. These are the cells, like in an organism. Let's take us as an example. Most of the cells inside of us are somatic cells. They're all the cells in our body other than the eggs and other than the sperm, and then any of the cells that give rise to eggs and sperm. You gotta understand, like boys, you have special cells in your body that will produce sperm. Those cells don't undergo mitosis. They undergo something else. And ladies, you have special cells in your body that make eggs. Those cells don't undergo mitosis either. So somatic cells are basically every other cell with the exception of the ones I just talked about. So examples of somatic cells would be skin cells, liver cells, pancreatic cells, those kind of things. Those are the kind of cells that are going to undergo mitosis. Okay. All right. Okay, here we go. Let's kind of go into the process. But before we go into the actual process of mitosis, remember I said that a cell actually spends most of its time in something called interphase. Remember, this was the crust of the pizza. This was the outer edge. And the only thing I want to mention here, and this is we're going to be drawing a lot of circles and pictures today, which is good. A really important thing happens in interphase. You have a cell here. You have a cell that has a nucleus, and inside that nucleus you have 2 chromosomes. I drew the chromosomes as little lines there. So obviously this is not a human cell. It only has 2 chromosomes. All of our cells have 46 chromosomes. 2 chromosomes. The really important thing that happens in interphase, again, we're not in mitosis yet, is that DNA, those chromosomes are going to replicate. In other words, they're going to go from that form that you see here on the left, and then they're basically going to turn into sort of what looks like this X shape. It's still 2 chromosomes over here. And in fact, I'm going to still write that. You still have 2 chromosomes on this cell on the right, However, those chromosomes basically made a copy of themselves and glued it on. This portion right here is identical to this portion right here. And down in the smaller chromosome, this portion right here is identical to this portion over here. It almost looks like there's 4 chromosomes, but they're not. There's actually 2 chromosomes, but they have like copies of themselves glued together. That's a sister chromatid. In other words, the chromosomes on the left over here are like single by themselves. But when those replicate themselves, when they make a copy of themselves, they turn into this X shape. That X shape is called a sister chromatid. The reason they call it that, let me kind of show you this picture here. I kind of just took, it doesn't really look so much like an X in this picture. They actually, I'll tell you what, I'll draw a better picture of it that looks more like, kind of like an X. Okay, that's probably a better picture, because that's the kind you see in the textbook. Either or are right. That's a sister chromatid, or I put or that. In other words, this one over here on the left is identical to the strand on the right. That's why they call them sister chromatids. It has nothing to do with being female. It just has to do with you have 2 strands that are identical. All right, you're going to have to see. Cells absolutely have to copy their genetic material in order to produce more cells. Think about it. If I plucked a cell out of you, and you have 46 chromosomes, and I said, all right, we need to divide up those chromosomes. We need to make more cells. You absolutely have to copy the material, or else you're going to not have enough stuff to put in each of the 2 new cells. That's why you have to have this stage happen. That's why you have to have that, that's why what you just drew, that's why you have to have them replicate themselves. All right. Moving on. Now let's talk mitosis. It is a 4-stage process. And those are the names of those 4 stages. Very, very, very important for you to know those 4 words and what happens during those 4 stages. Mitosis, prophase, is always the first phase. Metaphase is the second phase. Anaphase is the third phase. Telophase is the 4th phase. P-M-A-T. Remember that order, definitely. P-M-A-T. All right, so let's start at the beginning. Let's start at prophase. And what I want to do is just give you, hold on, I'm going to kind of go right along with you. I want to give you guys just kind of a brief overview of what happens at each phase. All right, hold on. I'm going to start with like a blank cell. Okay. 3 things happen here. 3 major things worth noting. The nuclear envelope is going to disappear, the chromosomes are going to become visible, and something called the mitotic spindle is going to form. So let me tell you about those 3 things here. I've got a cell here. And I'm going to draw a dotted line, a dotted circle on the inside. That dotted circle is going to be called, it's the nucleus, but I'm really more concerned not about the nucleus, but I want you to know that that's what I'm calling a nuclear envelope. It's just a fancy word for like the outer covering surrounding a nucleus. I drew it as a dotted line on purpose because it's in this phase where that thing is literally going to start disappearing. If you guys remember, what's inside the nucleus is your DNA or your chromosomes. And so I mentioned here, what happens is the chromosomes are going to become visible. Well, remember, up to this point in interphase, I told you that basically you're not going to be able to see chromosomes. It's all really thread-like. But it's at this phase where those chromosomes are going to become visible. And look, I'm already drawing them as X's. Remember, I'm drawing them as X's because they basically, they copied themselves just a short time ago, like you saw above. So they appear as sister chromatids. All right, this other thing happens. There's a really cool structure called a mitotic spindle. And the mitotic spindle, you kind of have to think of it this way. This is what I call the mitotic spindle. Think of like Spider-Man and how he likes to shoot webs out of his wrist or whatever. Well, you almost have to think of the mitotic spindle kind of like that, because basically you're going to need some kind of piece of equipment that is going to be able to attach onto these chromosomes and move them around. So you almost have to pretend like Spider-Man's kind of shooting out these fibers, these protein fibers. Those protein fibers are going to hook up with these chromosomes, and they're going to be able to kind of like move them around like puppets. That's what the mitotic spindle is. It is. It's almost, and it kind of looks literally like almost like a little end, and then you have these like webs shooting out of it. And it kind of forms on both sides. It's almost like these chromosomes are getting attacked from both angles. Again, this process of mitosis, you're going to want to move chromosomes all around the cell. Well, they're not just going to magically float there on their own. You absolutely need the mitotic spindle there to move the chromosomes. All right, so those are the three things that happen in prophase. That's all I want you to know. Next phase, metaphase, and it's easy to remember what happens at metaphase, because what's going to happen here are those chromosomes, and I just drew 2 chromosomes above. Those 2 chromosomes are going to line up in the middle of the cell, as I have shown here. There are 2 chromosomes lined up. They're lined up single file. In other words, they're not grouped. These 2 chromosomes didn't line up next to each other. They are literally single file in that cell. That's really, really important. How do they line up in the middle? Well, that mitotic spindle got them there. Okay, so I like to say that during metaphase, the chromosomes line up in the middle. All right, what's next is anaphase, and anaphase is also easy to remember because anaphase starts with an A, and so does the word away. And what happens in this stage is those chromosomes are going to basically get plucked apart from each other. The chromosomes are going to then move away from each other. That's why the word, it's easy to remember anaphase as being away, because they're literally going to separate and move away from each other. Those sister chromatids are going to separate. Prior to them being pulled apart, remember they look like an X, like you see here below. But anaphase literally rips that one away from that one, and that's why you see them going to their separate ends of the cell. You are ripping apart the sister chromatids. But remember, this one is the same as this one. So this is a really key step of getting the same genetic material in 2 different cells. Chromosomes move away from each other. The sister chromatids are separating. Okay. Got one more.
Can you leave that up there?
Yeah, I did that.
Long.
Okay. We good? Finally!
Finally, so done.
Telophase. Remember, P-M-A-T. We're at T. Telophase. You can start to see signs of 2 cells forming. Okay, now this is the last stage of mitosis. Remember, you still have just 1 cell though, but that cell is showing signs of forming into two cells. Here are the important things that are going to happen. You've just successfully put 2 chromosomes on one end of the cell, 2 chromosomes on the other end of the cell. Remember, the parent cell started with 2 as well. The nuclear envelope is going to reform. That's what that dotted line is. That's the nucleus, basically returning back. The nuclear envelope is returning back to normal. That mitotic spindle is going to dissolve away. They don't need it anymore. There's no need to move any chromosomes around anymore, so it just goes away. No sense of having it around. All right. Mitosis is done as of now. However, the process of cytokinesis, are we get to put that back?
Just the last one.
The process of cytokinesis is where you're actually going to get division of the cytoplasm.
How to spin the baton.
I'll have these notes available after class if you've missed something. I'd rather have you guys pay attention to what I'm saying and not be so concerned about writing it down. Remember, all these notes are available online as well. So I would rather have you pay attention to what I'm saying
I
and
just
not
like playing games online. I like studying with the games.
not desperate about copying down everything. If you miss something, you'll get it. Cytokinesis is the cytoplasm dividing in half. That's where you have 2 new cells. Okay, it's going to vary depending on if you're an animal cell or a plant cell. Plant cells and animal cells are very, very different. Animal cells. When animal cells undergo cytokinesis, literally what's going to happen is that cell, and I kind of drew it like that above, the cell's going to actually kind of pinch in the middle, and it's gradually going to keep pinching and pinching and pinching and pinching and pinching, and bam, you have 2 cells. Okay, actually, that pinching in is called cleavage, believe it or not.
I'm sorry.
No, that's fine. Everybody else has been laughing.
Immature.
Exactly, but that's what it's called, and that's the name of the process whereby the cell pinches inward to form 2 new cells. That's what happens in an animal. In a plant, totally different. See, plants, they have no change in shape. What happens in a plant cell is you have 2 ends of the cell. There literally is going to form, it's almost like they're forming a brick wall between them. That brick wall is called a cell plate. And that cell plate is going to kind of form between 2 cells. The cells actually aren't going to separate from each other. Plant cells are kind of different. They really kind of, they're kind of right next to each other. There's like no elbow room. But when you put up a cell plate there, it basically is going to, once it builds up enough, the 2 cells will act independently. So you have 2 cells. Okay, guys, those were the steps of mitosis. Very, 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 very important. Hold on, I want to put one picture up here.
Shh.
Are we done writing stuff down?
Okay. Mitosis summary. You know what, I want to kind of look at these one at a time so it's not so overwhelming. Okay. Look at the very top. At the very top of the page, you have a cell. You have a cell. Actually, it's not even it. This isn't even mitosis yet. This is interphase. You can tell that this is an interphase. Look at this picture right here. You have kind of the animated version of the cell. Look at the genetic material. It's all thread-like. You can't make out the individual chromosomes. You have no idea how many chromosomes are here. That's clearly an interphase here. You can see a cell. So you can see the nucleus has the dark matter in there. You can see that it's actually genetic material, but you can't make out any of the distinctive chromosomes here. That's interphase. All right, the next phase, prophase. Now we are in mitosis. Now we are in mitosis, and the key things, remember, that happen here are that you're going to start seeing these chromosomes. Look at this picture. You can start seeing, they almost look like little worms or whatever. You can see the individual chromosomes. The other thing that happens here is that, looking up in this picture, you can see this kind of border around called a nuclear envelope. You cannot see that here. It's starting to go away. You can't see the spindle in this picture, so I don't know. All right. 4 chromosomes, it looks like, is the story. But you can look at these chromosomes, even though you can't make them out here. Look at this picture. You see 1, 2, 3, 4 chromosomes here. Obviously not human. But the chromosomes look like X's because they basically have copied genetic material. So this, like on this red chromosome, that left side of that red chromosome is identical to the right side, but this is all still considered to be 1 chromosome. So 1, 2, 3, 4 chromosomes in here. Prophase. The next phase is metaphase. Remember middle, and you can easily see in this picture here that the chromosomes are all lined up in a line in the middle of the cell. And they show you a better picture over here. Metaphase, middle, they're lined up in the middle.
I understand.
Oops, anaphase. You do understand it?
Yep.
Good. Anaphase away. Look what happens here. I want you to look at this picture compared to this picture. You can see these chromosomes separate. They're going to separate. They're not going to be sister. They're not going to be X's anymore. They're not going to be sister chromatids anymore. They are basically being ripped apart at the center. 4 are going to go one direction. The other 4 are going to go in the other direction. Anaphase away. And telophase. Here, I can just take the picture off. Okay. Now, telophase over here. Your nuclear envelope is going to start reforming. You still technically are still 1 cell, although you're showing signs of not. And then finally, cytokinesis is where the cell is actually going to pinch off in this case and split into 2 new cells. Yep, this is actually, that was my next question. It's not a plant cell, this is an animal cell. Remember, the way that this pinches is the same as an animal cell. So you can tell this is an animal cell. All right, I want to show you a little animation, a little video. Yeah, it takes like 3 minutes.
Probably not.
I still got to copy down like half the slides. I didn't have your number. 46042. 46042. What? 46042. Is that really? That's what it's like really easy to remember.
It's like that.
428.
It's new in a few weeks.
Alright. Okay, guys. This is a picture. It's just like it's better to see mitosis and cytokinesis as it's actually happening. I'm going to actually show you this thing 2 times. The first time I'm going to let you listen to the narrator, and then the second time I'm actually going to speak through it. They're going to mention some words in here, some big words. So I don't want you to get, some words you've heard, but some words you haven't. So I want you just to kind of listen to her first, and then listen to me.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers.
Okay, now we're in mitosis.
Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle.
The yellow thing is the mitosis.
Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of 2 sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, I want to show it one more time, but the words were actually kind of big. I'm going to simplify it with kind of what we talked about in class.
This soccer player's knee will be as good.
Okay. Obviously, repair of cells is one of the key things mitosis and cytokinesis does. Again, we're starting an interphase. We're not in mitosis yet, but remember, a really important thing happens at interphase that I'll talk about in a second. But the first picture they show you, that go inside the nucleus, that purple thing's a nucleus. All those purple things are the thread-like DNA. Remember, in this phase, you can't even see the chromosomes. However, the really important step of DNA replication takes place during interphase, even in the thread-like form. They make a copy. You've just made it a sister chromatid. Those two strands are identical. Identical, identical. They're sister chromatids. And then they just said that they're made up of DNA and protein. And then still interphase, but it kind of starts a little bit to sort of kind of collect itself and condense. But it isn't until prophase where that's really going to happen. Now that yellow thing is that mitotic spindle I was talking about, the Spider-Man's kind of webs. That thing forms during prophase. And it basically forms and kind of surrounds the nucleus. Meanwhile, in the nucleus, now your genetic material is condensing into chromosomes. Now they're becoming visual. Now you can actually see them under a microscope. This and this strand are identical. Those are sister chromatids. This mitotic spindle, that yellow thing, is going to get into place, get into position. Those yellow fibers are going to hook up with the chromosomes. And then it kind of becomes a tug of war. They connect at a certain area, and they literally are going to just like tug of war, push and pull, getting those chromosomes to basically align themselves. So what's going to happen are these chromosomes are going to line up in the middle during metaphase with the help of that mitotic spindle. Remember, they're pretty much going to line up single file. Okay, they're getting ready to divide. But those sister chromatids line up single file. Then during anaphase, they're going to pull away from each other. So now those are going to separate, and they're going to migrate to the two ends of the cell. You've just successfully, basically, divided up the genetic material. Same genetic material on the left side of the cell and the right side of the cell. Same number of chromosomes. Telophase and cytokinesis kind of run into each other. But what happens, telophase, is you're going to see the nuclear envelope start to reform again. That's that purple ball thing. You got the nucleus coming back on both sides. And then it kind of runs right into cytokinesis, which is just the cell's actually going to split into two. So again, it was kind of interesting. You know, millions. This mitosis is going on inside your body right now. Okay, especially skin cells. They're pretty much the ones that divide. They're probably one of the fastest dividing cells. Your skin cells, which makes sense. So that's kind of that. Any questions on that process? Tomorrow, you are going to be doing a lab where you're going to look under the microscope at onion root tips. And you're like, onion root tips? By the way, the movie Twilight, I know it's older, but the movie Twilight.
Woo! They mention it in there from Belle and Edward's first meet, by the way.
Do you guys remember the scene?
Yes, I do.
There's been a scene where they are in science lab and they are looking at a microscope, passing it back and forth, and naming off phases like anaphase.
Can I bring it in and we can watch it?
No, but it gives you a reason to watch Twilight because that's exactly what they were doing. They were talking metaphase, they were talking anaphase. No, exactly. By the way, hold on. Remember at the very end they won the golden onion? Okay, they were looking at onion root tips, which is what you guys are going to be looking at tomorrow in class. Now you're going to be looking at slides that have already been prepared. Listen. So that's what you guys need to do. You guys literally you're going to be having a Twilight lab where you and your partner are going to be looking at a cell, figuring out what phase it's in, and getting to know the phases of mitosis really, really well. So that's it for tomorrow. You guys don't have school on Friday.
Jordan.
That's it. Oh, go ahead, Zane. You can
What was that?
turn the red one.
That person. The red.